video introduces the operation of transferring the cells in the bottle to the culture bag on the 7th day. On the 3rd day, we only need to carefully add an appropriate amount of medium and a certain proportion of plasma along the side wall of the culture bottle according to the instructions. On the 5th day, after transferring the cell suspension in the T75 bottle to the T175 bottle, we should rinse the T75 bottle as thoroughly as possible to collect as many cells as we can. After that, we can observe under a microscope whether there are still a large number of adherent cells. If so, we can add 20 milliliters of expansion medium and continue culturing. This additional medium will be added to the culture bag during the next medium replenishment. In this video, the stand on the right-hand side can facilitate our operation. If there is no stand, we can also hold the syringe by hand to operate. On the 7th day of cell growth, depending on the cell growth status, we usually add 200 milliliters or 400 milliliters of medium, that is, add fresh medium at a ratio of 1:1 or 1:2. We unscrew the long sampling tube and connect it to a 50 milliliters large syringe. First, add an appropriate amount of medium. The method of adding the medium is as shown in the video. We need to know how much medium we add in this step. Then, we pour out the remaining medium, about 50 milliliters, to rinse the remaining cells in the bottle to ensure that all the cells in the bottle are collected. Then, put the culture bag into the incubator for pre-warming. At this time, the total volume of the medium should be 200 milliliters or 400 milliliters. We take out the T175 bottle, gently shake it, not too violently to avoid breaking up the cell clusters. Then, pour the cells in the bottle into the culture bag through the syringe. As we can see, there is an additional 50 milliliter centrifuge tube on the left side of the stand. That is our plasma tube. On the 7th day, when replenishing the medium, we also need to add 1% plasma. We first add a small amount of medium to the culture bottle, then add an appropriate amount of plasma for rinsing, and then add the medium to the culture bag. Next, add the remaining medium in the centrifuge tube to the culture bottle for rinsing to ensure sufficient collection of cells. Finally, put the culture bag into the incubator for culturing. Since the volume of the liquid in the culture bag is relatively small, we suggest folding the bag in half for placement this time. It can be placed flat after the next medium replenishment. For the culture bottle, we can observe under a microscope whether there are still a large number of adherent cells. If so, we can add 20 milliliters of expansion medium and continue culturing. This additional medium will be added to the culture bag during the next medium replenishment.